Okay, everybody, now we are going to start Unit 6 material. So this is all about gene regulation, so turning genes on and off. And the first part of that is starting with DNA and understanding how that gets copied and transferred into each cell as they go through the cell cycle and meiosis, mitosis, all of that. So things you're going to need to know include the structure of DNA. So that's kind of pulling from freshman year stuff. I'm going to talk a little bit about the history of DNA and how we got to where we are today. I wouldn't say that you have to free memorize all of the names of people and their experiments. However, if you're presented with these, you kind of have to know how to apply them. We are going to learn that the replication of DNA is semi-conservative, and I'll describe what that means. And it always occurs in the 5' to 3' direction. Again, I will explain. You're going to need to know what all of the enzymes do in this replication process. So there's a DNA polymerase, ligase, helicase, topoisomerase, and so on. And notice how they all end in ase. So you should realize that ase means enzyme, and all of these will contribute to the process of replication. You're going to need to know the general difference between bacterial chromosomes and eukaryotic chromosomes. And then how we actually package it up differently, and depending on how it gets packaged, that can affect the ability to express those genes. Okay, so the first original problem that, historically we had was, what is the genetic material? So is it made of DNA, or is it made of proteins, or something else? There was a point when we did not know the answer to this. So way back, my God, almost 100 years ago, Frederick Griffith developed an experiment. So he took mice and he infected them essentially with bacteria, or a virus. So what he did was he had living S cells. So that's what these are. So live infectious pathogen, and then he would inject them into the mouse, and the mouse dies. Well, then they found that there was a non-viral pathogen that they could try. So then in this case, this is the negative control, whereas over here we had the positive control. We know this pathogen infects cells. We know that it will result in the mouse dying. So that's a positive control. The negative control is we're going to infect them with this pathogen that does not cause disease. And so in that case, they injected it into the mouse, and the mouse was fine. So again, negative control. So then what they did was they took the viral version, or the pathogenic version, the one that causes disease, and they heat killed it. So we know that heat can denature proteins. And what they found was that when they did this, the mouse lived. So that tells you that they have essentially murdered the pathogenic component. What they interestingly found was that if they mixed this heat-killed version with the non-pathogenic version, somehow the mouse still died, and there was a living pathogenic version inside that mouse. So what this says is that even though the proteins were eliminated, somehow it was able to pass the pathogenic or infectious component onto the normally not infectious component and kill the mouse. So what they found was essentially that the living R bacteria transformed. And we can talk about transformation at another point. But it's somehow that resistant bacteria was transformed into the deadly S bacteria by some unknown heritable substance. And then it took, about 15 years later to determine that upon testing of DNA, RNA, and proteins and various types of heat-killing experiments, that they then eventually figured out that transforming agent was actually DNA. And then a little bit later on, what they found was that there are, or I guess what they used, is bacteriophages. These are viruses that infect bacteria. And inside of them, they have DNA and proteins. And you can kind of see the picture of what that looks like. The DNA is inside the phage head, and then whenever it goes to attack a bacterial cell, it kind of injects its DNA into that bacteria. So then the bacteria can be like a little factory cranking out tons and tons of virus. So what they were able to do is actually label these different components. They labeled proteins that were made by the virus, and then they also labeled the DNA, and they could actually see where it went. And here's kind of what that looked like. So they radio labeled, or they used, in this case, sulfur to label and fluoresce the proteins. And they had the phosphorus DNA, or they used that to track the DNA. So in the top row, you can see that the protein is labeled. They did a whole bunch of stuff. They, got the virus to attack and so on. And what they found was that the protein itself was actually inside the liquid, that it wasn't inside the cells. And then here, when they did the same thing with DNA, they found that the DNA was actually inside the cells. So what I guess the conclusion would be, is that the infectious material that the viruses or bacteriophages are using are DNA, because that is what is injected into the cell versus the proteins of the virus are trapped on the outside. So DNA is what is entering the infected bacteria. So therefore, it must be the genetic material. Here's another instance of what that looks like. This is just a different photo that goes along with that same experiment. Okay, so then comes the second problem. Now that we know that DNA is the genetic material, well, what does it look like? So someone by the name of Chargaff, I guess you could call it too, in 1947 came up with specific base pairing rules. And hopefully you remember from freshman biology, there are bases that make up DNA. There are 4 letters: A, T, G, and C. They stand for adenine, thymine, guanine, and cytosine. And what he learned by looking at various species is that the DNA composition varies. There aren't always the same amounts of A's and T's and C's and G's in every single species, which kind of makes sense. But he did notice something interesting. Whatever percent A was always the exact same as T. And whatever percent G was always the exact same as C. So he was really the one who came up with the base pairing rules based on those percentages to say that A always pairs with T and G always pairs with C. They have slightly different structures. A and G are larger. They have 2 rings, and they are called purines. And T and C are 1 singular ring, and they are pyrimidines. You might see those vocabulary terms later. The only way that I remember it, and it's super silly, is I think AG of agriculture, and agriculture is pure. It's ridiculous, but I will never forget that A and G are purines, and then therefore the other 2 are pyrimidines. Okay, so then comes along Rosalind Franklin. So a couple years later, she worked under Maurice Wilkins, and she was an X-ray crystallographer. So she tried to basically use X-rays to take images of structures. So she was really the first one to get a photograph using X-rays of what DNA looked like. So that is roughly how she contributed to the idea of it being a double helix, because it kind of looked like an S in the picture. But of course, at the time, they didn't realize that X-rays were such a horrible thing, and eventually she died of cancer as a result. But her contribution then led to Watson and Crick, a few years later, actually coming up with the double helix as the model and building it. So that's where we get that twisted ladder structure. And they were also able to use Chargaff's rules to say that A always goes with T and C always goes with G. And of course, they won the Nobel Prize. Here you can see what that double helix looks like. So there's what we call the backbone, which are the sides of the ladder. And the 5' end is always the top portion or the first portion. The 3' end is always the bottom portion or the end. And that has to do with organic chemistry and how we number things. Really, it has to do with the blue here, the sugar, because we number the different carbons. And based on where we number them, the 3 is in the bottom left, and that's where the next phosphate and sugar group will attach. Whereas up here, the head is on the 5' end. So that's where it originates from, but you just really always need to know that all of the directionality is 5' to 3'. The backbone itself is an alternating phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And it's important to know that the sugar, you can see that it's a 5-ring sugar, is deoxyribose. So it's ribose because it's a 5-carbon sugar, but it's called deoxyribose because it is missing an oxygen that ribose has. So that's one of the differences between RNA and DNA, is that the D stands for deoxy because it's missing one. But that's the backbone. And then the rungs of the ladder are those bases. So those are the 4 different nitrogenous bases, or as you will see them referred to a lot. And that would be half of the ladder. And then there would be a complementary strand that's attached. So as I mentioned, based on Chargaff's rules, we know that we have these 4 bases. Again, you can see them. And then what base pairs with what? Based on the overall structure, just knowing that A and T has 2 hydrogen bonds. So the 2 sides of the rungs of the ladder attach to one another using hydrogen bonds. And you can kind of see what that looks like. Whereas G and C have 3. So it's actually a little bit easier to break DNA that has a lot of A's and T's than it does for G's and C's. So as I just mentioned, hydrogen bonds are between those base pairs, and they basically hold the 2 sides together. They're like a zipper. So when we get to the part of actually doing DNA replication, we have to break those bonds in order to pull the 2 sides of the ladder apart, so then you can replicate it. Based on how this strand is set up, the main one that you would talk about is in the 5' to 3' direction. So it is from top to bottom, and you can see that the one on the left is that way. The other one runs anti-parallel. Basically, that just means in the opposite direction. So it's flipped upside down. So its 5 to 3 is in the opposite direction. So again, DNA strands are anti-parallel. The 2 sides, one of them is kind of upside down compared to the other. An important thing to note is when we pack this thing together. So one super long DNA strand is not conducive to DNA replication. So what they have to do is we have to pack it close together so that way it can eventually condense into chromosomes and be copied and packaged up and moved around and so on. So DNA itself is a double helix. We also have these proteins. They're called histones. And the DNA itself is going to wrap around these histones in order to basically package it up. It's kind of like if you have a super long hose, and you know how we have those like boxes that you can roll up your hose cord in, and you just kind of crank a handle and it rolls up, and then you can pull it out when you need it in the summer. It's kind of like that. So it's an organized system. The histones kind of come together, and the DNA will wrap itself around those histones. And it creates this thing called a nucleosome, or essentially beads on a string. And you can look at it here in the top photo, but it's a string of DNA where at various segments you can see that it's been wrapped around histones. And then it can kind of pack itself up, so it gets condensed. Anytime you see the word nucleosome, that means that it's in that histone complex. And essentially, that's what this looks like. It's a bunch of them wrapped all together. And then we start looping them all kinds of crazy together. And then That's how we get a chromosome. So a chromosome really is a super, super compact DNA wrapped around tons and tons and tons of histones to kind of make sure that it's all connected together. Okay, so here's an important piece to understand when you're trying to compare the two. Perhaps there's a future FRQ question in your future of what's the difference between prokaryotic and eukaryotic DNA or something. Prokaryotic DNA is double-stranded, just like ours. But the difference really is in how it is organized. And we've learned this at the beginning of the year also. Prokaryotes, for some of you refresh your memory, prokaryote means before a nucleus, so bacteria, for example. They have one circular chromosome. That's it. It might be longer, it might be shorter, but it's one circle. And they don't have a nucleus, so it just kind of hangs out in the center of the cell. We have linear chromosomes, and as we've learned, we have 23 pairs or 46. Different organisms that are eukaryotic have different numbers, but the idea is that it's usually more than one. And because we do have a nucleus, all of our DNA lives inside of there. Prokaryotes supercoil their DNA. It's kind of like what we have, except there aren't really histones. It just kind of compacts itself really tightly and is called a nucleoid. They don't have those histones to wrap around. Whereas us, we have the spaghetti-like structure of chromatin, which is DNA wrapped around the histones. It's not super compact in a chromosome until DNA replication is about to occur, or I guess until mitosis is going to happen, but it is wrapped around histones. Okay, so then now that we know that the genetic material is DNA and we know what it looks like, well, how does it copy itself? So there were a couple different competing theories about how this could happen. One, it was thought that there was a conservative model. So you have your DNA and then that stays intact and you just make a bunch of just straight up copies. There was also something called a dispersive model where the idea was essentially the template or parent DNA was just chopped into tiny little pieces and then you end up with this hodgepodge mix at the end. And then the third, which is what ends up being actually true, is called semi-conservative. What happens is the original parent strand separates and you have two original strands in dark blue. And then you use that as a template to generate a new side of DNA so that the daughter or second replication is half from the parent, half newly made, hence semi-conservative. And then that process would continue and continue and continue. Meselson and Stahl are the main people that came up with this idea to understand. So they labeled bacteria with various things. They labeled it with a heavy isotope and a light isotope, and they basically wanted to see what would happen after they replicated and what was incorporated in or not. And based on that, they were then able to come up with the true idea of semi-conservative. So here's kind of what that looks like in another picture form. The daughter cells, again, have one half of the parent strand and then one that has been newly synthesized. Okay, this is a good picture of DNA replication. You might not actually know what all of these things are yet, but you will. We're going to go through them together, but this is a nice summary photo for you. Okay, so major players, holy enzymes, right? This is a good review of all of those different enzymes that you might want to star. So let me just preface this with enzymes are very often named by their function. And they end in ace, not 100% of the time, but a lot of the time, and all of these do. But the name of it gives you an indicator of what it does. So a helicase is one that unwinds the helix. It is the thing that actually breaks those hydrogen bonds in between the two sides of the DNA in between the nitrogenous bases to unzip or unwind. So helicase breaks the helix. And this process starts at what we call an origin of replication. And I'll talk about that in a little bit. Then initially some proteins are going to come in and separate the two strands, and they form this little magical kind of complex called the replication bubble, which we'll see later. There's something called topoisomerase. Topoisomerase basically allows for the DNA to be unwound to actually replicate. Because remember, the DNA is either supercoiled or it is wrapped around those histones. And you can't really separate the two sides of DNA if it's compactly unwound around histones. So topoisomerase is the thing that actually goes and unwinds them so that it is free to be broken in half by the helicase. And this happens ahead of the replication fork, which again, I will show you. And that's kind of where the progression of replication is going. So it unwinds it right before you get ready to copy it. Primase is the enzyme that puts down a primer. A primer in this case is made out of RNA, and it's targeted to the area that you want to replicate. So it's the very first thing that kind of comes in and initiates the replication process. It primes the DNA to be able to be copied. Then we have two DNA polymerases coming in. The first is DNA polymerase 3. And really the numbers just have to do with the order in which they were discovered. So it's silly, but 3 was discovered after 1, even though 3 works first. So DNA polymerase 3 is basically what we traditionally think about. Polymerase is something that creates a polymer. It is the major player that is moving along the DNA and pulling in the free nitrogenous bases, the A's, T's, C's, and G's, and adding them onto what we call the leading strand. And newly synthesized DNA is going to be made in the 5' to 3' direction. Again, I will show you what that means in a little bit. The leading strand is on the original template DNA. The other side is called the lagging strand. And the lagging strand is going to be adding segments in chunks called Okazaki fragments, which again, I will talk about. DNA polymerase 1 is an enzyme that takes off the RNA primer and replaces it with DNA. Again, we are doing DNA replication, so it doesn't really make sense that RNA is going to be in our finished product. So the DNA polymerase 1 basically overwrites the RNA primer with DNA letters. And finally, at the end, DNA ligates or joins the two halves of the ladder back together. So it reforms those hydrogen bonds once the replication process is complete. Okay, so let's go through some of these. Helicase unwinds the helix at the origin, and it creates what we call replication forks. If you think about like a fork in the road, I think that's where that's coming from. So we have a double-stranded DNA, and what happens is helicase comes in. Maybe we have an origin of replication where you want to start the copying process. So at these origins of replication, you can see helicase has come in and peeled apart the two DNA strands. The dark blue are the parent strands, and then the light blue will be the new strands. It's kind of going in both directions, so those are called the replication forks, the pushing point and the directionality of where the replication process is going to be. And then when all is said and done, you'll end up with 2 strands of DNA instead of 1. Primase is going to add the RNA primer. So again, here is a replication fork. And we have helicase, which is unzipping, essentially. It's moving this way. And I had mentioned topoisomerases previously, these are the things that are moving ahead of the replication fork and making sure things are unwound. So it's unwinding it so that the helicase can essentially then unzip it. And the next step is the primase coming in and adding an RNA primer. You can see here that this DNA strand is in the 5' to 3' direction, and this one is 5' to 3' in the other way. So DNA replication is going to actually occur from right to left on this top, from 5', because it's the opposite of what's it attached to 3' this way. And then in the red, you can see the RNA primer, and that will attract the polymerase to come in and start to actually add new bases. So, as I mentioned, DNA polymerase 3 is going to add nucleotides in the 5' to 3' direction. So here's the template. If this is 3' to 5', then the new one is going to be 5' to 3' here, and the polymerase is going to go in and add. So a new nucleotide triphosphate has the base and then 3 phosphates. And essentially what the polymerase does is it chops off 2 of those phosphates, and then the last one is used to join to the previous sugar. And that's how we end up with alternating sugar, phosphate, sugar, phosphate, sugar, phosphate. Here's what that looks like. So we have the RNA primer that was added to the added by the primase. And then there's a couple other accessory proteins helping on, but the major one here is DNA polymerase 3. And you can see it's bringing in those new nucleotides, and it's working in the 5' to 3' direction. And so here's what it would look like after it moved down a little bit farther. This is all taking place on the leading strand of DNA. In this picture, you can kind of see that in this case, this picture is a little tricky. The origin of replication is right in the middle here. And so what happens is replication is going to start at those black dots, and replication is going to move to the right, and a separate replication is going to move to the left. They kind of go in both directions at the same time. So depending on which direction you're going, you have to kind of take the leading strand and lagging strand into account. The primers are in red. So DNA is always added in the 5' to 3' direction. So for example, here, if this was being added in 5' to 3' direction, it's just one long continuous strain. However, there's a problem. The other strand is going the opposite way. So what they end up having to do is make these little chunks and then attach them in an overall 5' to 3' direction. Again, those are what we call Okazaki fragments, and we'll get to those hopefully in a little bit. Like right now. So these are short segments of DNA that are made in the 5' to 3' direction, and then they're tacked onto the lagging strand. So here's my template, and you have the primer. So the primase came in and added the primer, and you make a little fragment. And then you move down a little bit and you make another fragment that runs into the first. Then you move down again and make another fragment that moves first. Because what's happening is that overall, the direction of replication based on where it is really needs to go from right to left. So replication needs to go in the 3' to 5' direction. But DNA is never made that way. It's only ever made in the 5' to 3' direction. So it has to make this little chunk kind of going backwards, and then the polymerase jumps back and makes a little chunk, then jumps back and makes a little chunk. So each tiny segment is made in a 5' to 3' direction, but then they're glued together in an overall opposite. I know this is confusing. Okay, so here's kind of what that looks like when you put it together. This top parent strand is 5 to 3. The replication fork is moving, let's say, from right to left. So overall, things should be going from right to left. The top strand doesn't have a problem because the polymerase would be moving from a 5' to 3' direction. It's just one long, nice, continuous chunk. However, that's not the case for this lagging strand down here. It needs to overall go from 3' to 5'. So it makes these little Okazaki fragments, and you can see that it's kind of being chunked out together. And then ligase is the thing that comes in and basically glues them together. Okay, so the polymerase is the thing that brings in the new nucleotides, but it's also prone to making mistakes sometimes. So what they try to do is they proofread as they're going. They want to make sure they're not making any errors. So we have what's called mismatch repair. So there's enzymes that basically come in and say, nope, you messed up, and swap out the incorrect pair. So for example, if instead of A and T got stuck together, now you have a C and a T, which shouldn't happen. There's also called excision repair. So that would actually have a nuclease come cut out the damage, and then it basically has to copy over it again. So that would be for a larger scale problem. And that's actually what we do for CRISPR, for those of you who learned about gene editing. So here's kind of what that looks like. Oops. So if there's a problem, so let's say these were improperly paired together, there could be a nuclease that comes in and actually cuts out that chunk. And then polymerase will have to come in and recopy that area, and ligase would come in. So traditionally, when DNA is being copied, pairing errors occur about 1 in every 1,000 nucleotides. And this complete DNA thing happens in 1 in every 10 billion, which seems like not a lot. But if you think about the billions of base pairs that you have, it's fairly common. Okay, so let's see. Oops. Problems at the 5' end. DNA polymerase can only add nucleotides to the 3' end. So it in and of itself can never repair or complete 5' ends of the daughter strand. So essentially what happens is the RNA primer comes in and always starts every fragment, right? And then polymerase finds that primer and extends. And then polymerase 1 usually comes in and replaces the RNA with DNA. But it can't do that in the 5' area. Like, it can only add to the 3'. So what happens is over time, it gets a little bit shorter and a little bit shorter and a little bit shorter. So we have something called telomeres. And telomeres are at the end of our DNA. They are repeated units. They're like little caps, the end of our DNA. So that after it gets copied and copied and copied over and over and over and over and over again, it allows for those little snippets to be cut off without hurting any of the segments of DNA or the sequences that we need. So there's telomerase, which is an enzyme whose job it is to add those telomeres. And you can see them here, they've been stained orange. And oftentimes what happens is in cancer cells, those telomeres might be not there. And therefore you can get DNA damage. So here's what that looks like. The telomerase comes in and basically makes sure that there's that cap on there. It extends the 3' end of the DNA strand. The other strand is extended in the usual way by primase. So that part's not an issue. And so overall, you get this little like overhang. And those overhangs, again, are used to help protect DNA during the replication process. So that's pretty much the end of this particular presentation of DNA replication. You survived.